Buenas tardes desde la Escuela Española de Historia y Arqueología del CSIC en Roma. Bienvenidos nuevamente a esta nueva conferencia de nuestro seminario Online Talks. Seminario que siempre repetimos, realizamos junto a Miriam Cubas de la Universidad de Alcalá y Robson de la Universidad de York. Y como siempre, la parte técnica, agradecer la ayuda de Matteo Benati y en la organizativa a todos los compañeros del CSIC en Roma. Today the lecture is Professor Oliver Craig, and I let Miriam Cubas to meet.
Yeah, it's for me a pleasure to introduce our speaker for today, Oliver Craig, with which I have had the opportunity to work in several projects. Oliver is the deputy head of the Department of Archaeology at the University of York and professor of archaeological science at the same university. He's a specialist in biomolecular archaeology, with the recovering of proteins, lipids, and DNA from ancient skeletal remains and archaeological artifacts to provide scientific evidence of past human activities. His research has focused on the analysis of materials from key prehistoric sites in the UK, Central and Eastern Europe, and along the Atlantic, Baltic, and Mediterranean coastlines. He's currently working in important archaeological sites in Europe, from the Neolithic, like La Marmota, for example, and the Bronze Age, like Mass Farm. So thank you for this, Oliver, and thank you for your contribution to our seminars, and you have the word.
Thank you very much, Miriam, and thanks very much to the organisers, Juan and Harry as well. Yeah, a great seminar series. You can see that it's going down really well, and it's a pleasure to be invited and to talk to you today. So I'm just going to share my screen. So hopefully everyone can see that.
Yes, it's okay.
Right, okay, great. So I really thought this was the last kind of online webinar I'd be doing for a while, but it looks like, looking at the news, this is going to continue for a while. So here we go again. And what I wanted to do today was not really talk about my own research, but to, if you like, take the pulse of the field and to look at this important field, biomolecular archaeology, and trying to assess its impact, particularly on the studies of the transition to farming in Europe. So my talk is really looking at different aspects of biomolecular archaeology and seeing what impact they've made. And really, I hope this is just more of a point of discussion rather than anything else. Okay, so the first thing to note, I think, is that biomolecular archaeology is a relatively young field. Really, the origins can be traced back to, at the very earliest, the 1970s. And this is when you had the first basic lipid residue analysis of ancient amphora was published in 1976. The first kind of stable isotope analysis of bone collagen also dates to the 70s. One of the earliest studies was actually in 1981, when a researcher in Denmark was using stable isotopes to study actually the transition to agriculture. So that's looking at bone collagen to look at diet. The first reports of ancient DNA, so DNA from skeletal remains, was actually from an Egyptian mummy, which dates to 1985. So these were very kind of early days in the field at the time. These were followed by the 1990s, and it's difficult to think what happened in the 1990s to this field. We had a lot of excitement in the 80s. The 90s was more of a time of reflection. A lot of the earlier studies were questioned. A lot of the DNA results were thought to be unreliable. There were issues of contamination. No one really understood much about degradation. And the field went into a kind of reflective period where a lot of the early results were actually questioned. And it wasn't really till the 2000s that the field was rejuvenated by a whole range of different researchers who started to ask more kind of fundamental questions, questions about the methodology. There was a paper in 2000, which was Ancient DNA, Do It Right or Not at All. It laid down criteria for how to undertake ancient DNA analysis and how to report the results and really set the standard for work going forwards. There was work published in 2001 by Matthew Collins' group, which really started to look at the conditions for which molecules preserve in skeletal remains, pointing out that it's not just about how old something is, but also the kind of temperature that those remains have been exposed to. And then in 2004, perhaps the biggest paper came out was when we had a whole genome was published. So this is amazing work, really, when you think that the whole genomes were really being only published on modern human remains just a decade earlier. We had the first ancient genome published by Eski Williseth's group in 2004. In 2008, we had widespread testing of pottery to determine early dairying. So before organic residue analysis to look at the use of pots was limited to just a relatively few studies. And I think what Richard Evershed's group in the late 2000s was able to test a lot more samples, allowing a lot more inferences to be made. And from then, really the field has gone from strength to strength. We've had protein sequences distinguish sheep and goat using the Zooms approach, which some of you may know about. The discovery of proteins in dental calculus to look at diet, but also the oral microbiome. Papers were published in 2014. First population genomics papers. So this was really looking at genomes or deep sequencing of a lot of individuals and really looking at the population level, was published in 2015. And then in 2017, we start to see big movements in methods to enhance our ability to study stable isotopes from human remains, to look at diet through the application of single compounds, identifying amino acids. And then more recently, I think another big breakthrough that's really going to be important going forwards has been actually AMS dating molecules directly associated with artifacts, lipids on pottery, for example. So relatively young field, but one that's really, really taken off and grown in the last few years. So what has been more generally the impact of biomolecular archaeology to the field of studying the past? And some have said, like, it's a 21st century revolution in understanding the past, similar to the radiocarbon dating in the 20th century. So we know that radiocarbon dating really revolutionized archaeology in the 20th century. Some suggested that biomolecular archaeology has done the same for archaeology in the 21st century. However, we can also pick up a few criticisms of the field, and these have been made quite widely in the press and in the media, and also in scientific journals. For example, we know, perhaps it's true, that the specialist knowledge to actually undertake this research is contained in relatively few labs. And quite often it's driven by specialists, so molecular biologists or chemists, rather than archaeologists. So they're coming from very different fields. Quite often the data is perceived as difficult to interpret. The way it's presented sometimes is difficult to interpret and not so accessible to archaeologists and prehistorians. And there's quite often a drive to publish this kind of cutting-edge research in high-profile journals aimed at general scientists, rather than at the broader archaeological community. So it's the Nature and the Science papers. And some have said that this has led to oversimplified interpretations and conclusions. And it's true that quite often you get quite large headlines, people really trying to make the point in these large papers. I've got to be careful here because I'm co-author on some of these, but the Beaker phenomenon, the genomic transformation in Northwest Europe, you can see that sensationalizing the kind of research is one thing that we need to be kind of aware of. But on the other hand, it's also trying to sell these to these high-profile journals that really want to take those larger stories. And then, of course, the problem with that is that the media pick up on this, and you end up with silly headlines like this. So let me go back now and just think about a particular question in archaeology, a particular area of prehistory, which is the transition to farming in Europe, which is, of course, what really underlies this seminar series. So how has biomolecular archaeology transformed this particular question? Well, I don't need to go too much into what we already know about the transition to farming in Europe, but I'll just lay down a few ideas and a few principles, perhaps. Obviously, there's been centuries of research to look at this question, to look at these very early agricultural sites that are across Europe, to look at their date, to look at cultural affiliations between, for example, pottery styles, to look at the nature of the archaeobotanical and faunal assemblages, and then more latterly been to do a lot of radiocarbon dating to try and put this into a sequence. And we have this quite strong narrative now where we see that agriculture following domestication in the Near East follows different pathways out of Anatolia into the Balkans, through into Central Europe and up into Northern Europe, this kind of Danubian route, and this other route, which seems to follow the coastal Mediterranean route associated with the impressed cardial complex. I would also say that another form of Neolithization, perhaps doesn't involve farming, but certainly involves pottery, is also coming through the Baltic Sea and into Europe. These hunter-gatherers, the kind of complex hunter-gatherers, some people call them, who are also interacting against this hunter-gatherer farming zone to the north. So we have a fairly good idea about that. Less clear, perhaps, is what the impact of the Neolithic period was. So we have, I always go back to this diagram, which is kind of the conflict between whether the Neolithic represents just a simple change in material culture. It's an ideological change, essentially, and the actual economic change, i.e. the transition to farming, moving from hunting gathering to agriculture, is actually much more gradual and perhaps wasn't really properly established until we get to the Bronze Age, when you get more kind of agrarian societies. And certainly a range of British prehistorians have suggested this in the past. So you see a big change in the artifacts, you see a change in material culture, you see a change in ideology, but you don't see a change in the economy at least straight away. You start to see the vestiges of domestic animals and plants, but they're not the mainstay of the economy till later on. Hugely controversial. Other people will say, oh, that's ridiculous. How can you decouple material culture and economy? These things must be interlinked, and the change is immediate for both. Also thinking about how we can track prehistoric economies, material culture and people, and we can have different models for that. For example, the classic, which we're talking about here, is really that hunter-gatherers were replaced by farmers. So you have a hunter-gathering society that is totally replaced by farmers coming in, bringing in new ways of living, bringing in new ideology, but also bringing in the domesticated resources. And of course, the polar opposite of that is this idea of acculturation, where the hunter-gatherers are actually just adopting farming, but actually there's no real differences within the genetic structure of the people at all. Other models are kind of complex hunter-gatherers, where you have hunter-gatherers who continue, but they're adopting vestiges of the Neolithic world. For example, starting to produce pottery, becoming more sedentary, and we probably see that in the Mesolithic of the northern part of the continent, and acculturation of farmers, which is I don't think many people really talk about this, but this is where you have farming groups moving into an area, but actually not reverting back, but actually adopting vestiges of the hunter-gatherer economy. So maybe keeping some things like pottery or sedentism, but actually leaving agriculture and moving into hunting and gathering. And we can have all these different kind of concepts, and it's quite interesting that we can start to track these by looking at material culture, people, and their economic pursuits as well. So if we think back now to what were the key questions that were identified in the 2000s, I remember attending a meeting in London, I think probably in about 2003, organised by the Wellcome Trust, who had ring-fenced a lot of money for bioarchaeology at the time, and they were trying to get groups to put in large grants to win that money, mainly in Britain and Ireland. And they kind of asked us as a group of range of specialists, archaeologists and scientists, what were the main questions that need to be tackled using ancient biomolecular analysis. And these are the kind of questions that came up. I looked back through my notes, and it was the classic: to what degree was the transition to agriculture driven by population replacement? Quite that what we saw before. To what degree did diets change? When were secondary products introduced? So dairy, traction, and wool. So the whole idea of the secondary products revolution. And then more kind of profound questions, which I don't think we really have a good handle on at the moment, or will ever have a handle on, such as how did population sizes change? And that does affect a lot of things if you actually think about it. And then more kind of larger questions, such as what the effects of health was of the transition to agriculture, and this idea that actually moving from a hunter-gathering way of life to a more sedentary farming way of life was perhaps, to quote Jared Darwin, the worst mistake in the history of humankind. So let's have a look at some of these. So it was thought, and still is thought by many, that ancient DNA has all the answers. Well, does ancient DNA have all the answers? Well, in terms of looking at the spread of farming and the spread of farming ancestry, this was a nice kind of study that was done recently, where they took all the genomic analysis that had been performed up to 2021 across Europe. And they first, on the left, they mapped just the spread of agriculture based upon sites that contained domesticated animals and plants. So we can see that the story of the spread from Anatolia and the southeast of the continent up through Central Europe and across the Mediterranean holds true when you look at the radiocarbon dates, and we can see quite a detailed map that's been put together here. And of course, a lot of other people have done that as well. Then if you compare that with the map on the right, now this is really interesting, because this is human remains that have, so data of human remains that have over 75% Near Eastern ancestry. And you can see, so farmer ancestry, if you like, and you can see that these maps are quite comparable. Both look, broadly the same. Obviously, the map on the right has got a lot less resolution, but that's because there's far fewer human remains that have been analysed. And I guess the question is how these maps show discrepancy. Have we got the resolution, or will we ever have the resolution to actually see a difference between the date that farming arrives in the place and whether that's associated with the movement of humans, or conversely the opposite? Do humans move into an area, but actually farming, humans with Neolithic ancestry, i.e. farmers, move into an area, but actually that precedes the arrival of farming? And those, I think, are the really interesting points that could be brought out of this. But at the moment, we can see that, broadly speaking, we have a very clear answer to the question that farming is associated with a demographic process, i.e. the movement of people. If we look in a bit more detail, and this is human remains from the UK, these are a lot of sites that was analysed and published quite recently. And it's quite interesting to see that actually there's not that much variation between the different parts of Britain. So if we look at, for example, Wales or Southeast England, Southwest England, we can see that the percentage of hunter-gatherer ancestry within these individuals is roughly about 20-25%, between 20 and 30%, where the majority, 75% or so, is Anatolian Neolithic ancestry. And these are all early Neolithic human remains. What's interesting is that the proportion of hunter-gatherer ancestry doesn't decrease or increase really as we move into later periods, into the middle Neolithic or into the later Neolithic. And it's broadly similar to what we see in Iberia. It's greater than what we see in Central Europe, but we see this kind of similarity in the amount of hunter-gatherer and farmer ancestry in Neolithic individuals in Britain. And this suggests really that these people have moved into Britain already holding some hunter-gatherer ancestry. It doesn't really give us the impression that there's integration between hunter-gatherers and farmers occurring on the British continent. What might have happened that there was some integration earlier, perhaps where they've come from, and that those people who are carrying some hunter-gatherer ancestry have moved into Britain at that time. So fairly good spatial study. But of course, what's really interesting is the kind of regional differences we see when we look at the radiocarbon dates and the degree of hunter-gatherer ancestry in humans. So for the British Isles, as I said, that's fairly constant. We don't see any real change after the initial arrival of farmers around 4000 calibrated BC. We see a similar level of hunter-gatherer ancestry. Whereas in France, and we've got to be a bit careful here because the sites are taken from different regions and areas, but what seems to be happening is that initially, so if we look at these red dots here, with the very earliest Neolithic in France, earlier than Britain, of course, we see quite a lot of hunter-gatherer ancestry initially, and that starts to diminish as we move through the Neolithic. So this could be an interesting example where you have the adoption of certain elements of farming by hunter-gatherers, but slowly that's eroded as further migrants are moving in from the Near East, bringing greater genetic homogeneity. And completely the reverse seems to be happening in Iberia, where initially we see very low hunter-gatherer ancestry in Iberia, but that seems to increase as we go through time, suggesting that there's some later integration with local hunter-gatherers. So this is on the scale of different countries. I'm sure, and I'm pretty sure this would be true, if we move to a more local scale, we'd see different effects going as well. And I guess the only thing that's really limiting us doing that is the desires of researchers to do it. Why keep testing more and more humans once the big papers have been written? But that could be done. And of course, the limitations of the skeletal remains themselves. And we have things like changes in funerary practice, the movement, for example, between inhumations to cremations, that obviously will affect the availability of human remains for genetic testing. So certain things we won't be able to do anything about. But I can see that these kind of studies, these more kind of regional studies, are really interesting. Another really interesting way of using DNA data is to look at mobility. And this really works on the idea that communities that have low mobility are more likely to have a greater genetic distance between them because they're not in a network, they're not interacting so frequently. Whereas highly mobile populations are going to be more genetically homogeneous. They'll have less genetic distance between individuals because obviously they're moving around a bit, providing greater points of contact. And so you can do some kind of, this is a nice study that was done quite recently using this approach to look at correlation between space, time, and genetic distance. And you can see that some interesting differences emerge between different regions and different periods. And if we look at this mobility estimator, which is kind of the higher this is, the more mobile the populations are likely to be, we can generally see as we move from hunter-gatherers from the Pleistocene, Mesolithic, through the Holocene to the Mesolithic, and through to what we're calling Neolithic hunter-gatherers, which would actually be the hunter-gatherers in the north of the continent who are actually pottery producing. We can see generally a decrease in mobility through time, which is really interesting and maybe the opposite to what you expect, especially when you compare with the Neolithic period when people have got agriculture, where you may think that we'll be more sedentary, but actually in the Neolithic period, we see actually a higher mobility estimate. And of course, perhaps not surprisingly, that's even higher in the Bronze Age when we see these mass migrations into Europe. But perhaps giving us a different kind of understanding of how we really think about mobility during these periods. And the fact that, complex, so-called complex hunter-gatherers who are producing lots of surpluses, maybe less mobile than the earliest farmers who are moving in. And another aspect, of course, I probably don't need to go too much into this, it's made all the headlines, is the kinship. So in the previous studies, I'll just go back a few, when we did the study in Britain, we only analysed 1 or 2 individuals per site. And the reason for that was to try and look at this population level, this large-scale process. But what really has kind of opened up is testing a lot of individuals on any individual site. And this allows, obviously, our ability to start to look at kinship in Neolithic societies, and also by inference how societies are organized. So classic study of the Newgrange Passage tomb in the Bend in the Boyne in Ireland. And you can see what this study showed was really interesting, was that a lot of the individuals have high coefficients of inbreeding. They were generally very closely related. But really, this individual here from Newgrange 10, has definitely made the headlines, showing kind of really incestuous relationship. So this would be brother, sister, mother, son, daughter, father kind of level of inbreeding going on, which is very hard to explain. Obviously, not something that you would expect to see. And the only real ethnographic analogies for this are seen in these kind of very tight patrilineal royal family structures. And this is, by analogy, they're arguing that this is what's happening in this Neolithic society, that you have this very kind of patrilineal society headed perhaps by a god king. So, really interesting results that have come out of Newgrange. And I don't know if many of you, there's been a recent Neolithic seminars group where a lot of other data was started to be produced from other chambered tombs across Britain and different types of monuments, and showing similar levels of inbreeding between individuals interred together. So really interesting data starting to emerge that's giving us an insight into social organization. Okay, so moving from the kind of genetics and looking at the people, but now looking at the economic transition to farming. Of course, we have many ways of studying this. We can look at the archaeobotany, archaeozoology, genetic studies of crop diversity, a long history for these. What's nice about this, we have direct ways of analyzing this economic transition. We can look at stable isotope analysis of skeletal remains to see the kind of foods that people have been eating. We can also look at DNA and proteomic analysis of dental calculus. So that's the mineralized plaque on teeth, and that preserves food molecules in certain cases. And we can also look at the genomic evidence for dietary adaptation. So how we've adapted to digest certain foods. And I guess the classic for that would be lactose and the ability to digest that as an adult, which implies dairying economy. And then, of course, the final thing, which we'll talk about, is the material culture. How does that fit in there? It's another way looking, and has been applied to look at an economic transition, although I would argue that this is very much a more cultural sphere of interpretation. So classic paper published by Mike Richards et al. In 2003, and really shows very similar to what was produced back in the 1980s for Denmark, which was once you move to the Neolithic, and this is radiocarbon years BP, so it's uncalibrated, but this dotted line is representing the arrival of agriculture. So to the right, we have the hunter-gatherer individuals that have been analysed from Britain, and to the left, these are all Neolithic individuals. So after the arrival of farming, we can start to see a shift away from marine foods indicated by the y-axis, which is the delta 13C value, the carbon isotope that's in bone collagen. And the more positive this number is, the less negative, means more marine foods in the diet. And so you can see that as we look into the late Mesolithic, maybe I can try that first. I'm not sure that works. We can see there's a high amount of marine foods, and that changes immediately after we get into the Neolithic period. And that includes, interestingly, individuals buried on coastal sites. So even in the Neolithic period, you're buried on a coastal, you're buried near a coastal site, you're living near the coast, presumably, you have access to lots of marine foods. There's no real evidence that that was a large part of your diet or the economy. Sharp shift in diet at the onset of the Neolithic, turning your back on the sea to face the land. Some taboos have been suggested against fish during the Neolithic period. What is the reason for this sharp change in diet? Really interesting. One kind of query on this is whether this kind of bulk collagen approach, this is extracting the collagen out of your bone and doing the isotope, has the resolution to really say that people aren't eating any fish at all. And a really nice study by Janet Montgomery's group was published in 2020, where they looked at this site in Sunborough, Shetland, Neolithic site, very coastal, obviously. And this is the islands in the far north of Scotland, for those of you who don't know where Shetland is. And what they did, rather than look at the bulk collagen, they also looked at the tooth, and they sectioned the tooth. And so they can look incrementally at different points of this individual's life, and as they're growing up, it's too. And what they see is actually at certain periods, and this is roughly their age, along the bottom, you see a sharp peak in nitrogen isotopes, and also in the carbon isotopes as well, suggesting that this individual had access to marine foods. They were consuming for at least small amount of time in their life, quite a large amount of marine foods. This individual at the top, no marine foods. This individual at the bottom looks like, between the ages of 8 and 10, they were consuming quite a large amount of marine foods. So this is actually making us kind of question the validity of just saying, oh, marine foods was out, people weren't eating this, it wasn't part of the economy. Clearly, in certain cases it was, it's just not detectable when you look at the bulk collagen. And when you look at this individual's bulk collagen value, this individual B here, it's actually showing a terrestrial diet. So it's only by doing this you can reveal that. Quite interesting, marine foods weren't a mainstay of the economy. It might have been a strategic fallback food, especially if we think about how the process of maritime colonization might have occurred. People moving along these coasts might have needed to rely on marine foods to supplement their economy in times when crops fail, for example. Just very quickly to say that this is work we're doing with Maria Fontanel's. We can get a lot more detail on individual diet and start to pick up the kind of missing components by not looking at the bulk collagen anymore, the actual collagen molecule from bone, but moving to the amino acids. The amino acids make up collagen. And each of these amino acids has a different story to tell in terms of diet. They're coming from different foods. So by measuring those independently, we can get a much more accurate picture of diet. And a lot of work is going on in this area now, specifically for Neolithic individuals. And I think the nice thing is that we can actually go back to the collagen that was extracted over the last 20 years, take those collagen samples if they're still knocking about in people's fridges, and we can analyze the amino acids to look at this more, using this more high resolution approach. Okay, so that's diet kind of dealt with. Organic residue analysis of Neolithic pottery has been undertaken massively over the last 20 years. More than any other period, people have started to delve in to look at the contents of pottery during the Neolithic period. And there's been kind of two approaches that have been used. I'll just outline them here. One is to look for chemical biomarkers. So these are compounds that can reveal the presence of a particular product in a pot. And the classic we've been using a lot in York is biomarkers for fish. So we have a chemical, don't worry what it is here, it doesn't really matter, but that when we find that chemical, it can tell us that that pot was being used for processing marine or freshwater fish or other kinds of aquatic products. We can also use another approach, which is to measure the isotope values of different compounds released from the pot. And that gives us a more quantitative way of assessing what the contents were. We can start to separate different different products based on their isotopic criteria, such as fish, dairy products, porcine products, and ruminant products. And it also gives us the quantitative variable, because if we start to see mixing of these things, then we can kind of have a better understanding of how much of each of these was present in the pot. Obviously, it gets quite complicated, but it gives us a more quantitative approach. Okay. So I think the first thing to say, if we look at some data, sorry, let's get that one. Okay. The first thing to say, if we can look at some data, is that, and this is a huge amount of vessels that have been analysed. These are all early farmer pottery, so within several hundred years of the arrival of agriculture at any one point on this map, and hunter-gatherer. So these are in orange, okay, orange dots, and the blue dots are hunter-gatherer vessels. So obviously, for much of Europe, central, southern, and the Atlantic coast, we don't have any hunter-gatherer vessels, right? Because hunter-gatherers didn't produce pottery in these regions, whereas we do have hunter-gatherer pots, Zwifterband in the Netherlands, Tabula pots here, and moving into the Eastern Baltic, where we can do a direct comparison. And if we look at how many times we identify fish or aquatic biomarkers in these different sites that have been investigated, you can see that it's quite stark that the orange dots through much of Europe, south, central, western, and most of the northern, are very small. There was zero aquatic products identified in pottery from these periods. And that's not to do with preservation, because other lipids were present. It's just the fact that pots weren't being used for this process at this time. The exception, and that compares, sorry, with the hunter-gatherer pots, which generally do have aquatic products in. So large amount of aquatic foods being used in hunter-gatherer pots. So perhaps not surprising, you'd say, this is associated with the economy, very agrarian economy, not much fish present in hunter-gatherer site, in early farming sites, whereas hunter-gatherers are using a lot of fish. The exception is in the Baltic region. And quite interestingly, we do see, even when we move into the farming period, we see quite high amounts often of aquatic foods. So, for example, in Denmark, we see this transition between the Tabula hunter-gatherer pottery to these funnel beaker sherds, so the funnel beaker culture, agricultural communities from Northern Europe, producing very different pots, but actually using the vessels in a very similar way. And isn't it interesting that this is the only place that really happens? And this includes, by the way, coastal sites in much of the rest of Europe, where you would have thought marine foods would be accessible at least. And one reason for this would be perhaps that there's some knowledge of pottery use, even though the pottery vessels change themselves, is persisted across the transition to agriculture. So we see this cultural kind of culinary or cultural continuity going from the late Mesolithic to the early Neolithic. So what do we find in the Neolithic pots? Well, no surprise there. In the majority of cases, we're finding dairy products. Well, maybe not in the majority, but at any site, there's generally dairy products that are present. And there's a lot more work that's being undertaken at the moment by the Bristol group, so this story might change. But as far as we know, we're seeing in many of these early Neolithic pots, right from the earliest part of the Neolithic, dairy foods in the vessels. So this is telling us that indeed dairy was part of the Neolithic economy from the very earliest time. We're not looking at a secondary products revolution. We're not looking at something that happened later. From the very onset of agriculture and pastoralism, we're starting to see at least the use of dairy. The frequency of dairy may change, but actually at the very early Neolithic, dairy foods were being used. And this is quite clear in many of the sites that have been analysed. So Miriam's study, as some of you will know, has shown actually that the frequency of dairying does change between sites, and work that Miriam Cubas did when she was in York was testing a whole range of vessels from Atlantic sites in Iberia and also in France as well, and comparing these with work that was already done in the British Isles. Many of these sites, coastal sites, very few, well, zero fish identified, but daring was identified in many of these sites, but actually the frequency, how often we identify dairy products in pots, changes as you move from the southern sites in Portugal through Cantabria to northern France and into Britain. And in Britain, actually, you've got, in the northern Britain, you've nearly got all the vessels that have been analysed contain dairy foods, whereas down in Portugal, only a few. And this is interesting. Obviously, this might imply that dairying actually got more popular or got more frequent as the Neolithic progressed through time and moved into these northern regions. People have suggested it might be to do with the need for vitamin D, which is accessible from milk and less accessible from sunshine, which we tend to have less of up in the UK than Portugal, which is no surprise there. But and also it could be other things about how an advanced cattle economy, which was very set on dairying, needed to be established before the agriculture could actually move into these northern areas. But whatever it is, looking more at detail at the frequency of these residues over these larger scales produces some quite interesting results and should be the focus of future studies. Looking at now at a single site, so that we've gone from kind of like mega regional, to regional, and now down to a single site. And this is work that I did in Durrington Walls with Lisa Marie Shillito and colleagues, where we were looking at just a single assemblage with really tight radiocarbon dates, so we could actually get the majority of pots that we analysed down to the 24th century BC, which is very good in terms of chronology. So we're looking at 141 vessels, all dating to a very short amount of time. And here's what we found, and don't worry too much about the density thing here, but this is the criteria using to infer dairying, and you can see these are the different categories, and these are on the y-axis is the frequency of pots that falls into these different categories. And it's interesting that we're not just seeing a kind of normal distribution. So if you had access to dairy products, ruminant carcass fats, so animal fats from ruminant animals like cattle or sheep, and carcass fats from non-ruminant animals, which at Durrington Walls, this site must have been pigs, because loads of pigs, then you might, and it was all being mixed together in the pots, then you might see a kind of normal distribution. Yeah.
Sorry to interrupt. It seems to have frozen on YouTube. Is there any way you can just press exit and then don't leave the Zoom? Just press exit to reduce the screen size, and then try it again, and it should kickstart it.
Close the presentation and open again.
Okay.
Thanks.
You want me to open the presentation again? Has that worked?
No, in this moment, no.
Okay, I'll quit PowerPoint.
Close, 1 second. Yeah.
How's that?
Oh, just your face is on the screen, but bear with.
Yeah, I've shared now.
Okay, 1 second, I'll let you know. There's always something, isn't there?
Yes, 1 second. I think it's work now. 1 second.
It's okay?
1 second. Yes, 1 second, Oliver. 1 second. It's for C.
Juan, I can see it on YouTube. It looks fine.
Yeah, I think it's fine now. Yeah.
Yeah.
Okay, shall I carry on?
Yeah. Do you want to just start from this slide again? So
yeah. Okay. So
yeah. Cheers.
all
Sorry.
right. So, okay.
Thanks.
Sorry about that. I hope it's like technical problem with YouTube. I think it was fine on my computer, but it might have just been a problem with rendering it on YouTube. But so if we look at just one site, and I'm coming to the end of my presentation now anyway, and we look at a lot of pottery just from one site, we can start to tease apart some other kind of interesting facts, which really relate, I think, to culinary preferences rather than anything else. So the x-axis, as I was saying, shows you the different classes of food product that we can identify, and the y-axis is really the frequency of those. If everything was mixed together, we'd expect a normal distribution. We don't see that. What we see is this kind of lump. So certain pots have been used for dairy, whereas other pots are being used for a mixture of ruminant and non-ruminant carcass fats. Now that's interesting, because what it's suggesting to us is that people are actually using certain vessels for a particular product, right? So they're choosing particular pots. And in this case, we're able to also see that these are thinner walled vessels, also deposited in certain parts of the site, are being selected for dairy production, whereas other pots have just been, meat has been put in, different types of meat are being processed together. So we're starting to see some culinary choice that's been exercised in the past. So this is really, I think, a very important kind of area to investigate further. And it's thinking about, rather than economy, what we're looking at is cuisine. And cuisine is a set of socially situated food behaviors. And really, I'm saying it should be a cultural attribute rather than an economic indicator. And it's inherited in similar ways, perhaps, and this is what we really want to test, to knowledge of how pots were made, or how artifacts were made, or how people were buried. It's a cultural attribute. And what's interesting, if we look at an archaeological site and we see that plenty of different foods are available, right? And then we look in the pottery, oh, sorry, wrong one. We look in the pottery and we only see that certain foods are actually being processed in the pots. That's telling us something. Certain foods have been excluded from pottery, certain choices have been made, fish are being processed in a different way. Whatever it is, it's giving us an understanding. And I think that's important in this context of the transition to agriculture, because essentially the transition to agriculture is about food choices. It's about how people decided which foods that they wanted to consume. Okay, so that's that. Missing evidence. This is a problem with organic residue analysis. We miss a huge amount of evidence. What is that? It's the plant products, the cereals, the fruits, the plant oils, and even the narcotics. We know that these kind of plant materials are present because we have excellent archaeobotanists who have been working.
Sorry, all the screen's frozen again.
Right.
I
don't
want
me to stop. I've got like 2 slides left. I can jump to the end.
Yeah, I mean, if you're happy to.
Or do you want me to just relaunch it one last time?
Relaunch it one final time. Give it a go.
Yeah, it's nothing I'm doing, is it?
I don't think so. No, I think it's due to the projection from.
I've
Zoom.
actually got it on a different. Let me just see if I can do this right. Sorry, I'm going to try and share it through Google. Maybe that fixes it. Maybe it's just Microsoft.
I can see your face. I just, yeah.
Can you see the screen now?
Just bear with, maybe. There's a slight time delay. 1 sec.
Yeah, now I think it's okay, Oliver.
It's okay, Juan.
Yes, I think.
Yeah.
I don't know what is the problem. Is it your connection or our connection? I don't know.
Yeah, I don't know. Is that okay? Can you see?
Yeah. It's okay now.
Right. Okay. I'm in a different program now, so it might work. I'm using Google rather than Microsoft. So plants, big problem. Cut a long story short, we do have ways of actually looking for biomarkers for these kind of materials. And cereals is one area. where a lot of research interest is happening. We've identified these in very well preserved cases. So this was a barrel from the Alps, but using molecular evidence for identifying the use of cereals, so not just the archaeobotany, but how those cereals were actually being processed on a site, what foods they were being mixed with, I think it offers a really exciting way forward. What about alcohol? Well, interestingly, you may know that wine has been identified or been reported to be identified in Neolithic ceramic vessels. Examples from the Caucasus, where they have used this biomarker called tartaric acid, and which is present definitely in these vessels. However, the interpretation of this has been somewhat marred, not least because this molecule is also present in many other types of fruit. So it's difficult to be so specific to wine. Perhaps a better case is later in northern Greece, in Dikili Tash, where you've got the vessels with the biomarker, the tartaric acid, but you also have good botanical evidence showing the presence of grape pips that have been processed. More recently, we've kind of worked with Lea Drieu to develop a method to kind of distinguish more reliably the presence of wine against other types of fruits, using the percentage of tartaric acid against a different type of fruit acid. And that seems to be a lot more reliable than just using the presence of tartaric alone. So really what we want to do now is a lot of wider testing. This study was actually done on amphora, but a lot of wider testing of Neolithic sherds for looking for the presence of wine. And my last slide on a residue analysis is the need to move beyond these approaches to what we can kind of call paleofoodomics approach, a kind of metabolomic approach, which is really using computer algorithms to look at all the molecules that are present within a pot sherd and trying to distinguish these using multivariate analysis to come up with a lot more high resolution analysis. So we're far from reconstructing recipes. This is an amazing cuneiform recipe, the oldest one in the world, from Iraq for pigeon and vegetable stew, possibly. I don't think we'll ever get to that kind of level using this approach, but we are making a lot of progress in mixing and understanding the mixing and separation of foods. What foods were put together in a pot, either at the same time or consecutively. And we need to think about the sampling strategy, deploy quantitative and qualitative approaches. I haven't had a chance to talk about the protein approaches, but that's one of those, and integrate with different types of culinary evidence. So to finish with, I think it's about time. How has our questions changed in the 2020s since the 2000s when we first asked this? Well, I think one of the big ones is now I think we are in a position to kind of tackle how the transition to agriculture changed social organization. I think we're in a much stronger position to look at the environmental, economic, and cultural factors that actually drove migration. We've accepted that migrations happened in the past. That's not a question anymore. Now it's understanding what are the factors that cause those migrations. And again, what was the role of hunter-gatherers in facilitating the transition to agriculture? Quite often they're silent actors in this story, and a lot more focus on their role needs to be thought about. For me, the presence of plant products in Neolithic diets and in pottery and culinary practices, there's a whole missing area there that we need to kind of explore. A lot of focus on animal products. We need to move to plants. And then also understanding how this knowledge of food production and consumption was transmitted, specific patterns of how people were producing and consuming foods, which we can kind of map and understand in a lot more detail. And of course, what we really need is more explicit spatiotemporal models, more regional studies, and of course, going back to the first point, greater interaction with archaeological investigations, which I think is starting to happen, and pretty optimistic about the future. So thank you for your attention, and that's it. I'll stop sharing. Okay.
Great, thanks all. Thanks for bearing with. If it's not me being thrown out of Zoom, it's something else happening. Anyway, yeah, great talk, great overview. I've got some questions then. You state perhaps we should be looking further at the spatio-temporal models, should I say, but yet perhaps there's some underlying criticism, not necessarily through your talk, but elsewhere as well, whereby we are generally looking at these big data sets and looking at the big picture. But do you think we kind of also need to look at smaller scale studies, not necessarily studies, but data sets themselves, and or say, look at it on a site-by-site cases again, kind of strip back these huge data sets and look at them from a more local scale?
Yeah, you're right. I mean, yeah, and there is a need for more regional studies, right? So, I mean, I did say that, and I think that is beginning to happen. It's whether people are going to invest the time and money in those kind of more local, regional questions, and whether we're going to see a next wave where all these advances that have happened already and have been applied to these big questions are actually rolled out and taken up and applied more widely to these kind of local situation. In terms of the spatio-temporal approach, that is still relevant to the local, because if you think about it, if you obtain radiocarbon dates from a site, and then you're trying to, so you have a site chronology, but then you're trying to build that into understanding, you know, the arrival times of different things and what's available at the time, then you need to have an overview of that bigger picture. I don't think that, I mean, what's been done is very good for Europe. I mean, best in the world, you could argue, tracking the spread of plants and animals, but it is done as a whole. So it tracks the Neolithic. We don't look at certain elements of the Neolithic. We don't decouple this, well, unpack the package, if you like, and look at the arrival times or of different elements of the Neolithic and how that may vary across time and space. And for that, we probably need a lot more, you know, higher resolution spatio-temporal models that are kind of artifact or product specific. Difficult to do, but I think that's kind of in the offing as well at the moment.
It certainly answers that question. I've got a question from Juan actually in the chat. He says, wonderful presentation. A question: if amino acids provide more correct results, or let's say high resolution results, are traditional isotope studies therefore still valid? I mean, you could say that they are, but basically the data is then being reanalyzed.
It depends on the question you're asking, right? I mean, so if your question you're asking, was there a big change? Let's take this one. If there was a big change in time with the arrival of agriculture, was there a big change, dietary change? The bulk isotope analysis kind of answers that question. You can see there's a gross change in the isotope signals as you move from the Mesolithic to the Neolithic. So something's happened, there's a change. If you want to know more precisely what to quantify that change, so was it 100% reduction in marine fish and 80% terrestrial animals, 20% cereals, then we can't really do that with bulk. Bulk doesn't have the resolution to really answer that kind of question. We can see a change with the bulk, but we can't actually be more specific about what was causing that change, what the new darts actually look like. So it depends. And if one of your questions is, did people abandon marine foods, then clearly I would say that the bulk isotopes aren't really sufficiently set to answer that question, because they can't really rule out that low level of consumption. And I think using the compound specific approaches, we might be able to do that.
So I've got another question, then I'll ask one myself, and then maybe we can end, because nobody else has got any questions, and we work with you closely anyway, also it kind of doesn't really need it. We don't really need to ask these questions. But my question was, where do you see us going in the future? I know you made these points at the end, but what sort of big questions do you think we will be able to answer in the future? Or is it just a case of not necessarily regurgitating what's already been asked and answered, but providing a different...
I mean, it's incredibly exciting time if you think about it. I mean, if you're an prehistoric archaeologist sitting here at the moment and you're thinking about what tools you have available to you at the moment, it's incredibly exciting. So the whole, the kinship studies are just in their infancy. Looking at larger amounts of human remains, looking at the radiocarbon dates, trying to understand the structure of those societies, how those individuals are related to each other, are there connections between different places within a landscape? I think it's going to really be absolutely revolutionary in the next 10 years. I think we're going to learn so much more about Neolithic society than we've ever really known before, and it's already happening now. The limits, as I said, are going to be, what's available, the human remains that are available. And then for me, for the economy kind of question, it is trying to understand the nature of that Neolithic economy and how it varies in time and space. We know that, domestic animals and plants are available, but how are those products being used? What proportion of, is it pastoralism that they're relying on? What happens to cereal agriculture? How important does that get established? How does it vary through time and space? Looking at how areas where it's more optimal for growing crops, how much is consumed by looking at these high resolution diets, and how plant foods are being used in conjunction with animal foods, and through looking at the pottery. I think that's a really exciting question for the future as well. As you might hear with Amy's talk tomorrow.
I've got one final question, which is just coming on the chat. So this person, I can't see the first name, but Meption, they've asked, I'm perhaps considering a lipid residue approach in my thesis on funnel beaker communities. However, I'm considering a perspective on climate change after the Holocene climatic optimum. Therefore, what is your opinion on farming communities returning to a more hunting-based food economy when the climate becomes colder? And what parameters would you use to investigate this? And lastly, how would a change in food economy be felt among the communities in the rest of Northwestern Europe? And do you know any people in the field who are investigating these issues?
Well, you can answer that one, Harry. You are. But climate is something we didn't really talk about, and climate has got a story to play, I guess, in this bigger tale. And yeah, reversions is one of the questions we like to think about. When agriculture arrives, see it in parts of Scandinavia, and then people revert back to hunter-gathering way of life. And an easy way to think about that there is correlating that with climate. But I think for that, we really need, and one thing what we suffer from is that we have very kind of detailed, well-dated climate chronologies, but we kind of trying to tie the archaeology into those is sometimes quite difficult. And trying, I think one approach that's really exciting is to try and get climatic records directly from things like human remains and artifacts. And for example, a direct answer to that for the funnel beaker question would be to try and get deuterium isotope analysis of, say, fatty acids present within the sample, which will actually give you an independent idea of what the actual environmental conditions were like. Enhanced radiocarbon dating of artifacts as well will be important, so you can tie it in. But trying to reconcile these different records is something that a lot of people are trying to do. It's really important. So yes, good question. Very interesting.
Very good question. Yeah, as you say, I think that is something we are kind of currently lacking, at least with these data sets we've been playing around with, is tying in, as you say, this climate change and or environmental change into the archaeological change. Good. Right, thanks all.
No problem.
Shall we end it there? Miriam, Juan, unless you guys have any further questions.
Yes.
I don't have a lot of questions, I'm sorry. But I think I have an impression, a suggestion maybe, or something like that. I think that we are in a perfect moment, because we have generated a lot of data. And we have a lot of different data from the archaeological, through archaeological, archaeobotanical, organic residue analysis for the neolithization processes in Europe. So I think for the future, the good, or maybe a good scenario would be just to try to combine all of them and to do a very nice, not just the big stories, but also the regional case of study and even the archaeological case of studies, because I think some archaeological sites in Europe are relevant enough to propose a big story, just focus on them. So I think maybe now our work should be just to think about new hypotheses and new proposals about these neolithization processes in different scales, you know, in different, I don't know, maybe not in big geographical areas, but maybe taking some specific archaeological sites and to try to work with all the data and the integration of all the archaeological data together.
Yeah.
So we'll see.
Yeah, and I think, you know, defining the scale of what we should be working at is kind of a major issue that we need to get right over the coming, you know, years. And a lot of that would need, what will have to happen is that the research is kind of democratized.
Yeah.
So it's taken away from just a few players who are leading it all and calling the shots to a kind of bottom-up approach where, you know, people who haven't got access to all these resources can start asking the questions and then start to connect with these labs. And I think that has to happen. And that will all depend on whether, you know, everyone wants to play the game and whether the cost and the price of analysis is lowered enough for that to happen. But I think all the signs are looking good, and it's quite exciting times.
Yeah, I agree with that. So yes, if we don't have more questions, thank you all for your inspiring talk. And thank you everyone for listening to us. And see you tomorrow, I think, because our next talk is tomorrow.
Yeah, tomorrow is the presentation of Amy Bogart, okay? She will present the conference, I look for, From Ancestry to Farming Practice, Understanding the Neolithic as a Process in Europe. And it's in the same time, 4 o'clock time, Italy time, sorry. And see you tomorrow, if possible.
Okay, see you tomorrow.
Yeah.
Ciao, Oliver.
Ciao.
Thank you very much, John.
Ci aspettiamo a Roma.
Sì. Al prossimo.
Ciao.
Ciao.